Good evening, guys. It's 6 already, so we can start the session. I'll share my screen. Please let me know if everything is visible and if I'm clearly audible. Is my screen visible? Can someone unmute and let me know if my screen is visible and if I'm audible? Can anybody let me know if my screen is visible? Is my screen visible? Guys, is my screen visible? Guys, can someone just unmute and let me know if my screen is visible? Okay, I believe my screen is visible. So I'll start the session for today. So good evening, everyone. My name is Aishi, and today it is the week 8 of the live session where we discuss some sample problems from last year and some of the assignments that you have already submitted. And we'll discuss some of the problems, and hopefully that will help you, gain a further insight into the course, which is Applied Environmental Microbiology, and that will help you solve problems in the upcoming future. And I'm a PMRF research scholar, and I'm also an NPTEL TA for this particular course, Applied Environmental Microbiology. So today's eighth live session. My eighth live session. There's another live session going on in parallel on Tuesdays, I believe. Today is my eighth live session, and we'll move ahead with our um, sample problems for today. So if at any point my screen becomes invisible, like my screen stops showing, or if at any point I become inaudible, please let me know so that I can reconnect the internet, whatever issue is happening. So moving on to the first question for today. Which of the following is the correct statement about the Winogradsky column? It is a device used for measuring pH in water samples. It is a model system that simulates microbial ecosystems in soil. It consists of mud, water, and carbon source layers. It is a tool for studying the behavior of macroinvertebrates in aquatic environments. Which of these four do you think is the correct answer? Anybody? I guess these have been covered in the course lecture videos, but anyway, we'll be discussing what a Winogradsky column is. Anybody willing to answer? At least attempt the question.
I think option 2 and 3 are correct.
Option 2 and 3 is correct. Okay. Anybody else? Anybody else? Okay, so the correct answer will be. Sorry, is it not working? Yeah, the option 3 is definitely correct. It consists of mud, water, and carbon source layers. Before discussing about option 2, let's talk about what a Vinogradsky column actually is. So, a Vinogradsky column is a simple device for culturing a large diversity of microorganisms. And it was invented in the 1880s by Sergei Vinogradsky, hence the name Vinogradsky column. And the device is basically a column of pond, mud, and water mixed with a carbon source, such as newspaper, like containing cellulose, or blackened marshmallows, or eggshells that will contain calcium carbonate, or a sulfur source, such as gypsum, calcium sulfate, or egg yolk. And incubating the column in sunlight for months results in a aerobic or anaerobic gradient, as well as a sulfide gradient. And these two gradients promote the growth of different microorganisms such as Clostridium, Desulfovibrio, Chlorobium, Chromatium, Rhodomicrobium, and Begiashoa, as well as many other species of bacteria, cyanobacteria, and algae. So the column provides basically numerous gradients depending on the additive nutrients from which the variety of the organisms we talked about they can grow. And the aerobic water and the anaerobic mud or soil phase are one such distinction. So because of the oxygen's low solubility in water, the water quickly becomes anoxic towards the interface of the mud and water. And anaerobic phototrophs are still present to a large extent in the mud phase, and there is still capacity for biofilm creation and colony expansion, as you can see in the images over here. And algae and other aerobic phototrophs are present along the surface and the water of the upper half of the column. So basically, when we talk about the construction of this Vinogradsky column, it is a rough mixture of ingredients, and exact measurements are not really very critical or crucial. There's a tall glass, approximately 30 centimeter long, greater than 5 centimeter wide, and it is filled one third full of pond mud, omitting any sticks, debris, and air bubbles, and supplementation of approximately 0.25% weight by weight calcium carbonate and 0.5% weight by weight calcium sulfate or sodium sulfate is required. And it can be given by ground eggshell or egg yolk respectively, basically, which are rich in these minerals. And it can be mixed with some shredded newspaper, filter paper, or hay for cellulose, basically. An additional anaerobic layer of unsupplemented mud brings the container to 2/3 full. And alternatively, some procedures call for sand to be used for the layer above the enriched sediment, as to allow for easier observation and sampling of the resulting populations. So this is followed by water from the pond to saturate the mud or sand and occupy half the remaining column. And the column is sealed tightly to prevent evaporation of water and incubated for several months in strong natural light. And after the column is sealed tightly, the anaerobic bacteria will develop first, including such as the Clostridium species. And these anaerobic bacteria will consume the cellulose as an energy source. And once this commences, they create CO2 or carbon dioxide that is used by other bacteria, and thus the cycle begins. And eventually, color layers of different bacteria will appear in the column. At the bottom of the column will be black anaerobic H2S hydrogen sulfide dominated zone with sulfur reducing bacteria. The layer above will be green sulfur photosynthetic anaerobic bacteria. Then the layer will be purple, which is sulfur anaerobic bacteria, followed by another column of purple anaerobic non-sulfur bacteria. And at the top there will be a layer of cyanobacteria, which are sulfur oxidizing bacteria. And this top layer of aerobic bacteria produces O2 or oxygen, which feeds back into the column to facilitate the further reactions. So while the Vinogradsky column is an excellent tool to see the whole communities of bacteria, it does not allow one to see the densities or the individual bacterial colonies, and it also takes a very long time to complete its cycle. However, its importance in environmental microbiology should not be overlooked, and it is still an excellent tool to determine the major bacterial communities in a sample. So, the prokaryotic bacteria and the archaea will exhibit an astonishing metabolic diversity, which far exceeds that of animals, plants, fungi, or any other higher organisms. And the prokaryotes literally keep our biological world turning by recycling all the mineral elements necessary for life support. So, two famous microbiologists pioneered the study of these processes: Sergius Winogradsky and Martinus William, I think, Bejering. And in contrast to the pure culture studies of other pioneer microbiologists such as Louis Pasteur and Robert Koch, these workers studied the relationship between different types of microorganisms in mixed communities. So this is nothing but a simple laboratory demonstration which illustrates how different microorganisms perform their interdependent roles. The activities of one organism enable another to grow and vice versa. And these columns are complete self-contained recycling systems driven only by energy from light. So these columns are easy to set up with a glass or perspex tube about 30 centimeter tall and 5 centimeter wide diameter, and mud from the bottom of a lake or water is supplemented with cellulose, which is newspaper, sodium sulfate and calcium carbonate, then added to the lower one third of the tube, and the rest of the tube is filled with water from the lake or river, and the tube is capped and placed near a window with supplementary strip lights. And all the organisms are present initially in low numbers, but when the tubes are incubated for 2 to 3 months, the different types of microorganisms proliferate and occupy distinct zones where the environmental conditions favor their specific activities. And this is how the idealist, Winogradsky column basically looks like. And the large amount of cellulose that is added initially, it promotes rapid microbial growth, which soon depletes the oxygen in the sediment and in the water column. And only the very top of the column remains aerated because oxygen diffuses very slowly through water. And that is what happens over here also. And the only organisms that can grow in anaerobic conditions are those that ferment organic matter and those that perform anaerobic respiration. As we have already discussed in previous classes, that fermentation is a process in which organic compounds are degraded incompletely. For example, yeast ferment sugars to alcohol. Anaerobic respiration is a process in which the organic substrates are degraded completely to CO2 but using a substance other than oxygen as a terminal electron acceptor. Some bacteria respire by using nitrate or sulfate ions in the same way as we use oxygen as a terminal electron acceptor during respiration. Some cellulose degrading Clostridium species also start to grow when the oxygen is depleted in the sediment. And all the Clostridium species are strictly anaerobic because their vegetative cells are killed by exposure to oxygen, but they can survive as spores in the aerobic conditions. And they degrade the cellulose to glucose and then ferment the glucose to gain energy, producing a range of simple organic compounds like ethanol, acetic acid, succinic acid, etc. as the fermentation end products. So this is what happens. So this tube was filled with sterile nutrient medium containing sulfate and organic acid and an iron nail, and it was inoculated with a pure culture of disulfo-vibrio, and after 10 days the contents turned black. So disulfo-vibrio has small comma-shaped cells. So this shows figure C shows a species of Theocapsa, which is basically a purple sulfur bacteria from the Winogradsky column, shown in this figure A, basically at the top of this column, and this has been viewed by phase contrast microscopy, and the purple color can be seen when the cells are very dense. The sulfur reducing bacteria such as disulfo-vibrio here in B can utilize these fermentation products by anaerobic respiration using either sulfate or other partly oxidized forms of sulfur such as thiosulfate as the terminal electron acceptor, generating large amounts of hydrogen sulfide by this process. And like in our own aerobic respiration, we use O2 and reduce it to H2O. So here the H2S or the hydrogen sulfide will react with any iron in the sediment, producing black ferrous sulfate. And this is why lake sediments and our household drains are frequently black. However, some of the hydrogen sulfide diffuses upwards into the water column where it is utilized by other organisms. And the diffusion of the hydrogen sulfide from the sediment into the water column enables anaerobic photosynthetic bacteria to grow, and they are seen usually as two narrow brightly colored bands immediately above the sediment, a zone of green sulfur bacteria, then a zone of purple sulfur bacteria. And the green sulfur and the purple sulfur bacteria gain energy from light reactions and produce their cellular materials from carbon dioxide in much the same way as plants do. However, there is one essential difference. They do not generate oxygen during photosynthesis because they do not use water as a reductant. Instead, they use H2S, and the equations show that in parallel it will be 6 CO2 plus 6 H2O, which will give rise to glucose plus 6 O2, which is basically plant photosynthesis. Or it can be bacterial anaerobic photosynthesis, which will be 6 CO2, 6 carbon dioxide plus 6 H2S, hydrogen sulfide. It will give rise to glucose and 6X. So the purple sulfur bacteria typically will have large cells and they deposit sulfur of granules inside the cells. And the organism shown here is a species of Theocapsa. The green sulfur bacteria have smaller cells and typically deposit sulfur externally. And the sulfur or the sulfate from it produced by the photosynthetic bacteria returns to the sediment where it can be recycled by desulfur vibrio, part of the sulfur cycle in natural waters. Now, most of the water column above the photosynthetic bacteria is colored bright red by a population of population of purple non-sulfur bacteria. These include species of Rhodopseudomonas, Rhodospirillum, Rhodomicrobium, and a mixed culture of them can be shown in the bottle here in figure E. And these bacteria grow in anaerobic conditions, gaining their energy from light reactions but using organic acids at the carbon source for cellular synthesis. So they are termed as photoheterotrophs. And the organic acids that they use are the fermentation products of other anaerobic bacteria such as the Clostridium species. But the purple non-sulfur bacteria are intolerant of high hydrogen sulfide concentrations. So they occur above the zone where the green and the purple sulfate bacteria are found. So the column shown at the top of the here have passed the stage where these organisms are common because the water columns become oxygenated by cyanobacteria. This is the figure D. Figure D shows cyanobacterial filaments which are predominated in the upper region of the column. Column 2 basically over here. Now many microorganisms can grow in the oxygenated zone at the top of the water column, but these distinctive types are of specific interest. There can be types like number 1, any hydrogen sulfide that diffuses into the aerobic zone can be oxidized to sulfate by the sulfate oxidizing bacteria, and these bacteria gain energy from the oxidation of hydrogen sulfide and they synthesize their own organic matter from CO2. So they are termed chemosynthetic organisms or chemoautotrophs. Similar types of organisms occur in soils, gaining energy from the oxidation of ammonium to nitrate, which then leaches from the soil and can accumulate in water supplies. Then photosynthetic cyanobacteria can also grow in the upper zones. These are the only bacteria that have oxygen evolving photosynthesis like that of plants, and in fact there is a very strong evidence that the chloroplasts of plants originated as cyanobacteria or the ancestors of the present day cyanobacteria that lived as symbionts inside the cells of a primitive eukaryote. Similarly, there is equally strong evidence that the mitochondria of present day eukaryotes were derived from purple bacteria. And once the cyanobacteria start to grow, they can oxygenate much of the water. So this happened in column 2 over here. The whole water column was dominated by a mass of cyanobacteria composed of spiral filaments, which is this one. These are the spiral filaments. Also, the top of the water column can contain large populations of sheathed bacteria. Now, these are aerobic organisms which use organic substrates but are unusual because as the bacterial cells divide, they synthesize a rigid tubular sheath from which individual cells can escape and swim away to establish new colonies. And many empty sheets are then seen in older colonies, and they are made up of a complex mixture of proteins, polysaccharides and lipids that are thought to protect the cells from predation by protozoa. The sheets also can be encrusted with ferric hydroxide, giving a yellow or rusty appearance to the colonies. So these are narrow sheets of the okay, I have not added. There should be another image which shows the sheath bacteria from the upper yellow orange zone of the Vinogradsky column. So the total summary is that this Vinogradsky column is all in all a very classic demonstration of the metabolic diversity of prokaryotes. All life on earth can be categorized in terms of the organism's carbon and energy source. Energy can be obtained from the light reactions, which is photoautotrophs, or from chemical oxidations of organic or inorganic substances, is a chemotropes, and in the carbon for cellular synthesis can be obtained from CO2 autotrophs or from preformed organic compounds, that is heterotrophs. And combining these categories we get the four basic life strategies: photoautotrophs, example plants; chemoheterotrophs, example animals or fungi; photoheterotrophs; and chemoautotrophs. And only in the bacteria and among the bacteria within a single Winogradsky column do we find all four basic life strategies. So it's a classic demonstration of how microorganisms occupy highly specific microsites according to their environmental tolerances and their carbon and energy requirements. And finally, the column enables us to see how mineral elements are cycled in natural environments. We focused mainly on sulfur, but there are equivalent cycles for nitrogen and carbon and other elements. So that is all about Winogradsky column. Moving on to the next question. What is a sequential batch reactor used in wastewater treatment? Anybody?
Option 2.
Option 2, to treat wastewater in batches. Anybody else? Any other option? So, what is a shell or sequencing batch reactor? So, yes, you're correct. It is used to treat wastewater in batches. So, what is the sequencing batch reactor basically? These are nothing but industrial processing tanks for the treatment of wastewater, and these reactors treat wastewater such as sewage or output from the anaerobic digesters or mechanical biological treatment facilities in batches. Sorry, oxygen is bubbled through the wastewater to reduce biochemical oxygen demand and chemical oxygen demand to make suitable for discharge into the sewers or for use on land. So while there are several configurations of sequencing batch reactors, the basic process and flow chart is similar. The installation consists of at least two identically equipped tanks with a common inlet which can be switched between them. The tanks have a flow-through system with raw wastewater influent coming at one end and treated with water effluent flowing out the other. So while one tank is in settle or decant mode, the other is aerating and filling. And at the inlet is a section of the tank known as the bioselector. This consists of a series of valves or baffles which direct the flow either from the side to side of the tank or under and over consecutive baffles, and this helps to mix the incoming influent and the returned activated sludge, beginning the biological digestion process before the liquor enters the main part of the tank. So this is how sequencing batch reactors actually work out. Function of sequencing batch reactors. So yeah, it is used to treat wastewater in batches. Moving on to the next question. Which of the following steps is not a part of the microbial community fingerprinting? Anybody? You could take your time, think about it, and then respond.
Option 4.
Option 4. Anybody else? Which of the following steps is not a part of the microbial community fingerprinting? Microbial or bacterial community fingerprinting? DNA extraction, PCR amplification of 16S rRNA gene, sequencing of the PCR products, isolation and cultivation of microbial cells. So the answer is option 4, correct. But what is microbial community fingerprinting or bacterial community fingerprinting? So bacterial community fingerprinting refers to nothing but a number of cultivation independent methods that are used to measure biodiversity in environmental samples or to profile microbial community structures. Example, in response to experimental conditions. Examples include denaturing, gradient electrophoresis, temperature gradient electrophoresis, terminal restriction fragment length polymorphism, and automated ribosomal intergenic spatial analysis. So these molecular techniques have in common that they determine the variants of a certain gene, often the small units ribosomal RNA, and use this measurement as a proxy for the actual microbial cell abundances in the sample. And it is therefore assumed that each gene variant, which is apparently a band or a peak in the fingerprint, corresponds to a certain microbial taxon, often referred to as the phenotype or the operational taxonomic unit. Not phenotype, phylotype or the operational taxonomic unit. So this is common schematic. So the community fingerprinting analysis starts from environmental samples such as soil, sludge or water from which total DNA is extracted. The target gene or region is then amplified using PCR and the resulting applicons are separated according to the melting behavior and the applicon length or fragment length after titration with the restriction uh, enzyme, and the output is then visualized using a two-dimensional gel image or an electrophoregram generated by the automatic sequencer. So why are these techniques of microbial fingerprinting very relevant? So fingerprinting methods are used to provide an overall view of the microbial communities, indications of microbial diversity and insight into the types of metabolic process. Sorry, someone is trying to join. Metabolic processes. Sorry. So what happens? This basically this gives an insight into the metabolic process occurring in the aquifer, that is notably the terminal electron accepting processes such as sulfate reduction, and some can be used to determine a subset of the microorganisms present in the sample. And this capacity is relevant and important because biodegradation inherently depends on the types and abundance of microorganisms present in the subsurface. For example, microbial fingerprinting methods can identify when adverse conditions such as low pH, either natural or following a remedy, results in low microbial biomass and microbial diversity, uh, rendering biodegradation unlike other under existing conditions. And similarly, microbial fingerprinting methods can be used to determine whether the overall microbial community has responded or recovered to remedial actions, while other EMDs are much more appropriate to detect and quantify known contaminant degrading microorganisms. Several microbial fingerprinting techniques can be used to identify the predominant organism present in the sample and to basically describe the microbial community. So what does this microbial fingerprinting do? So microbial fingerprinting methods are a category of techniques that differentiate microorganisms of groups of microorganisms or groups of microorganisms or groups of microorganism based on the unique characteristics of a universal component or section of a molecule. For example, phospholipids, DNA or RNA. And microbial fingerprinting methods provide an overall profile of the microbial community, and some can be used to identify subsets of the microorganisms present. And the types of microbial fingerprinting methods described below here include phospholipid fatty acid PLFA analysis is also there, denaturing gradient gel electrophoresis DGGE, then terminal restriction fragment length polymorphism TRFLP. So these are also known as genetic fingerprinting methods. Microbial fingerprinting methods have been used to investigate microbial communities at many different environmental remediation sites, ranging from metal contaminated sites to retail gasoline stations. And microbial fingerprinting methods can also have potential applicability in the other environmental monitoring efforts, such as tracking of fecal organisms, so-called microbial source tracking, identifying and tracking microorganisms potentially related to chemical compounds or originating from industrial or related activities, And how are the data used? So data generated from the microbial fingerprinting methods are used to understand which microorganisms are present and how they're intrinsically coupled to their experimental conditions, environmental conditions. For example, geochemical conditions such as the availability of electron acceptors, which influence which microorganisms are present and active at a site, while the microbial activities such as electron acceptor consumption can slowly impact the site geochemistry. So microbial fingerprinting method therefore can provide valuable information as to whether subsurface conditions are conducive to bioremediation, and in evaluating the effectiveness addition of an amendment to stimulate the growth and activity of specific groups of microorganisms capable of performing desired process. And microbial fingerprinting methods can also be used to track the overall changes in the microbial community over the time or in response to remediation activities. So data gathered from the microbial fingerprinting needs, then can be used to evaluate the performance of a certain bioremediation strategy. So that is the importance of microbial fingerprinting and why it is still very relevant. Moving on to the next slide. What is the main objective of using enumeration techniques in environmental microbiology?
option 2,
To estimate the number of microorganisms present in a sample. Anybody else? Anybody would like to try? Anybody else? Okay, so option 2 is the correct answer. This technique is used to estimate the number of microorganisms present in a sample. But what do you mean exactly by bacterial enumeration? So basically, when you have a party, it's important to ensure you have enough, chairs and cups for all of your guests. Enumerating is a very fancy word for counting, and this term can be a very fancy, but the main purpose is very simple. So they are large enough to see with your naked eye. However, sometimes things aren't so easily visible. Bacteria are microscopic single cell organisms that cannot be seen without a high powered microscope. However, bacteria are incredibly important for human health. Some bacteria live in harmony with our body, helping us to carry biological process like digestion. Other bacteria can be pathogenic and cause disease in humans. Either way, it's important to keep track of who's around in our environment and in what quantities. For example, scientists need to know how many bacteria are present in certain food and drink items to determine if they're sanitary or not. So, one way to do that is via bacterial enumeration or while counting bacteria basically. So, however, since bacteria are very small, so we can just look around to do the count. Scientists use a variety of methods to do this, which we'll basically learn about here. So there can be viable or total cell counts. So before we dive into the methods, it's important to check about two types of enumeration that scientists might use. For example, first, sometimes scientists just want to know about the viable cell count. So this is the number of cells that are alive at the time of counting, and these cells are able to grow, divide, and carry out their functions. However, some other times scientists want to use a total cell count, which counts all cells in a sample, whether they are living or dead. Different methods use viable versus total cell counts, and we'll basically look at the advantages and limitations of each. So now that all of that is out of the way, let's take a closer look at the different enumerated methods that are used by scientists one by one. So there is the standard plate count. So bacteria are microscopic, so it can be difficult to simply count them by looking at the sample. However, luckily, bacteria form colonies when grown on solid agar media, and these colonies are collections of cloned bacterial cells, all originating from one single particular cell. And the standard plate count takes advantage of this situation. In the standard plate count, a sample of bacteria is diluted, and the basically it is later plated on agar media, and the plates are incubated and then the colonies form. and the colonies can be counted with the naked eye, and then the total number of bacteria present in the sample can be diluted from the dilution factor. And since this count only takes into consideration bacteria capable of cell division, it produces a very viable cell count basically. Then there's a turbidimetric measurements. So the standard plate count, however, can take some time since the bacteria need to grow and form colonies. A faster way to ensure or enumerate bacteria is the turbidimetric measurements. So this method of enumeration measures the amount of light that can pass through a suspension of bacteria. The more bacteria, the less light that will pass through. The amount of light that passed through is measured by a spectrophotometer. Since this method only measures the amount of light passing through the sample, it produces a total cell count rather than a viable cell count. However, the spectrophotometer only measures how much light passes through the sample. How do we correlate that with the amount of the bacteria already present? Prior to measuring an unknown sample, same controls must be used to create a standard curve. So, using either direct counts or standard plate count, samples with known numbers of bacteria are used in the spectrophotometer, and an upward, basically standard curve is created, and then the amount of light that passes through unknown samples can be fitted on the standard curve, and the bacteria can be enumerated. Then there is another direct microscopic counts. So although bacteria can't be counted visually with the naked eye, you can see them under a microscope. Direct microscopic counts are great for looking at total cell counts. So this is because it involves using a known volume of liquid and thus being able to control the total number of bacteria in 10 to 20 microscope fields. So using the average number of cells per field, the total number of cells can be calculated using the total volume of the sample. And although this method allows scientists to directly count the cells within a sample, it can still be very time consuming. So these are before that, so as I was saying, this is basically the success of oil spill bioremediation depends on one's ability to establish and maintain conditions that favor enhanced oil biodegradation rates in the contaminated environment. And there are, you can find numerous scientific review articles that have covered various factors that influence the rate of oil biodegradation. So one important requirement is the presence of microorganisms with the appropriate metabolic capabilities. And if these microorganisms are present, then optimal rates of growth and hydrocarbon biodegradation can be sustained by ensuring that adequate concentrations of nutrients and oxygen are present and that the pH is between 6 to 8 or 9. The physical and the chemical characteristics of the oil and the oil surface area are also very important determinants of the bioremediation process. So there are two main approaches to spill bioremediation. One is bioaugmentation and one is biostimulation. So in bioaugmentation, what happens? In this case, well-known, oil degrading bacteria are added to supplement the existing microbial population, and the other is biostimulation, in which the growth of the indigenous oil degraders is stimulated or induced by the addition of nutrients or growth limiting co-substrates. So this is a question of biostimulation. So definitely you're not adding the question asks which is not an example of biostimulation. So in case of biostimulation, we do not inject bacteria, rather we add particular nutrients and we try to induce the growth of indigenous oil degrading bacteria. We stimulate the growth of bacteria, we do not add bacteria ourselves. So that is why injecting bacteria, this option is correct here, because this is not an example of biostimulation. You are correct. So, if we talk about the difference between bioaugmentation and biostimulation, bioaugmentation is the process of adding specific microorganisms to enhance the existing populations and promote the biodegradation process, whereas biostimulation is the process of adding electron acceptors, electron donors, or nutrients to stimulate naturally occurring microbial populations in the contaminated area. And here we can see biostimulation is uh, related to indigenous microorganisms. Indigenous means very like related to that particular environment. We are not taking microorganisms from somewhere else and adding it to the system. We are adding certain nutrients or certain factors that will stimulate the growth of the indigenous microorganisms. So the microorganisms that are growing in case of biostimulation are from that particular place itself. Whereas in case of bioaugmentation, we exogenously supplement with microorganisms. So we take cultured microorganisms and we put it in the system to help degradation of the oil. So that is how these two are different. So in case of bioaugmentation, you can see there are cultured microorganisms that are cultured in labs or taken from other zones, and they're exogenously added to that system to help in the degradation of the oil spill. So these are some of the drawbacks. The introduced microbe often cannot be established in the environment, and these introduced organisms rarely survive within the environment. Since we are taking cultured microorganisms and we are adding it to the in the wild to basically help them degrade so that they can degrade oil. Sometimes these cultured microorganisms don't really have it in themselves to survive in the wild, and sometimes they get depleted, they do not survive in the wild. But here in this case, the drawback, there is another drawback in case of biostimulation. Due to contaminant toxicity, the existing microbial population may not be enough for the biodegradation process. So in this case, since the number of microorganisms is not in our hand, because we are not adding microorganisms ourselves, we are inducing or triggering or stimulating the growth of indigenous microorganisms. Here the existing contaminating toxicity, the existing microbial population that will happen might not be enough to curb the contaminant toxicity. So these are the differences between bioaugmentation and biostimulation. So you were correct. The injecting bacteria into oil spill will be the correct answer, because it's not an example of biostimulation. So moving on to the next question, acid mine drainage is a significant environmental concern associated with mining activities. Which of the following is a primary contributor to the formation of acid mine drainage? Anyone like to try?
sulfide minerals.
Anybody else? Yes, you're correct. So, we talked about acid mine drainage in some of the previous classes also. So, mine drainage is found basically when pyrite or an iron sulfide is exposed and it reacts with air and water to form sulfuric acid and dissolve iron. And some or all of this iron can then precipitate to form the red, orange, or yellow sediments in the bottom of streams. Basically containing mine drainage, and the acid runoff further dissolves heavy metals such as copper, lead, and mercury into groundwater or surface water, and this is very toxic, and the rate and degree by which acid mine drainage proceeds can be increased by the action of certain bacteria. So problems associated with mine drainage include contaminated drinking water, disrupted growth and reproduction of aquatic plants and animals, and the corroding effects of the acid on parts of the infrastructure such as bridges. So the most direct and immediately obvious toxicological effect of the use of coal has been acid mine drainage. And acid mine drainage is caused by the oxidation of metallic compounds such as the mineral constituents of rocks and solids and soils that is often present in coal mine slag. And most streams affected by the coal mine drainage are acidic, like pH 2.5 to 6, with high iron and sulfate concentrations. Also, ferric hydroxide often precipitates as a fine flock that may coat stream bottoms and further harm aquatic life. So, acid mine drainage can also leach toxic metals such as copper, aluminum, and zinc from rocks and soils. Copper is particularly very toxic to fish. And the oxidation or weathering of pyrite and other metal sulfides is ongoing as minerals are exposed to air, and these reactions may be sped up by bacteria. For example, a number of streams in the southeastern United States are naturally acidic due to the surrounding pyrite rock formations. So, the potential for a mine to generate acid and release contaminants is dependent on many factors and is very, site specific. So, Ferguson and Erickson, they are two people. They identified primary, secondary, and tertiary factors that control acid drainage, and these factors provide a convenient structure for organizing the discussion for acid formation in the mining environment. Primary factors involve the production of acids such as oxidation reactions. Secondary factors act to control the products of the oxidation reactions such as reactions with other minerals that consume acid. Secondary factors may either neutralize acid or react with other minerals. Tertiary factors refer to the physical aspects of the waste management unit, example, pit walls, waste rock piles or tailings impoundments that influence the oxidation reaction, migration of the acid, and consumption. Other downstream factors change the character of the drainage by chemical reaction or dilution. These downstream factors are beyond the scope of many papers that have been published online, and there are it's not discussed properly. And primary factors of the acid generation include sulfide minerals, water, oxygen, ferric iron, bacteria to catalyze the oxidation reaction and generated heat. Now, some sulfide minerals are more easily oxidized, such as framboidal pyrite, marcasite, pyrohite, and hence may they have a greater impact on the timing and the magnitude during acid prediction analysis compared to other metal sulfides. Also important is the physical occurrence of the sulfide mineral. Like well crystallized minerals will have smaller exposed surface areas than those that are disseminated, and both water and oxygen are necessary to generate acid mine drainage. So water serves as both a reactant and a medium for bacteria in the oxidation process. Water also transports the oxidation products, and a ready supply of atmospheric oxygen is required to drive the oxidation reaction. Oxygen is particularly important to maintain the rapid bacterially catalyzed oxidation at pH values below 3.5. Oxidation of sulfides is significantly reduced when the concentration of oxygen is in the pore spaces of mining waste units is less than 1 or 2%. So different bacteria are better suited to different pH levels and other edaphic factors. Edaphic factors pertain to the chemical and physical characteristics of the soil and water environments. So the type of bacteria and the population size change as their growth conditions are optimized. So some of the scientists have actually identified some of the bacteria that are involved, in catalyzing the oxidation reactions and their growth conditions. And the oxidation reaction is exothermic with the potential to generate a large amount of heat and therefore thermal gradients within the unit. So heat from the reaction is further dissipated by thermal conduction or convection, and these things like There's a popular research by Lu and Zhang on waste rock using stability analysis that indicates that the convective flow can occur because of the high porosity of the material, and convection cells formed in waste rock would draw in atmospheric air and continue to drive the oxidation reaction, and convection gas flow due to oxidation of sulfide minerals depends on the maximum temperature in the waste rock, and the maximum temperature depends on ambient atmospheric temperature, strength of the heat source, and the nature of the upper boundary. And if the sulfide waste is concentrated in one area, as is the case with encapsulation, the heat source may be very strong. So there are other secondary factors also. Some secondary factors uh, can act to basically either neutralize the acid produced by oxidation of sulfides, or may change the effluent character by adding metal ions mobilized by the residual acid. So neutralization by the acid by alkalinity released when acid reacts with carbonate minerals is an important, means of moderating acid production. So the most common neutralizing minerals are calcite and dolomite. And products from the oxidation reactions, the hydrogen ions, metal ions, may also react with other non-neutralizing constituents. And possible reactions include ion exchange on clay particles, gypsum precipitation, and dissolution of other minerals. And dissolution of other minerals contributes to the contaminant load in the acid drainage. Some examples of metals occurring in the dissolved fluid can include aluminium, manganese, copper, lead, zinc, and various others. Similarly, some of the tertiary factors affecting acid drainage are the physical characteristics of the material, how acid generating and acid neutralizing materials are placed, and waste and the hydrologic regime in the vicinity. The physical nature of the material, such as particle size, permeability, and physical weathering characteristics, is important to the acid generation potential. And particle size is where a fundamental concern, since it affects the surface area exposed to weathering and oxidation. And surface area is inversely proportional to particle size. Very coarse grained material, as is found in waste dumps, exposes less surface area but may allow air and water to penetrate deeper into the unit, exposing more material to oxidation and ultimately producing more acid. And air circulation in coarse materials is aided by wind, changes in the barometric pressure, and possibly convective gas flow caused by heat generated by the oxidation reaction. In contrast, the fine grained material may retard air, and very fine material may limit water flow. However, finer grains expose more surface area to oxidation. So the relationships between particle size, surface area, and oxidation play a very prominent role in acid prediction methods. And as materials weather with time, particle size is reduced and exposing more surface area and changing physical characteristics of the unit. And even though very difficult to weigh, each of these factors influences the potential for acid generation and are therefore very important considerations for the long-term effects of acid mine drainage. So sulfide minerals is definitely one of the primary contributor to the formation of acid mine drainage. So you're correct. The answer will be sulfide minerals. Okay, moving on to the next question. Which of the following statements about acclimation is correct? Anybody?
Option 4.
Anybody else? Anybody else has any different options? So, what is acclimation, and what is the difference between acclimation and adaptation? So, you are correct. Acclimation is the process by which organisms adapt to changing environmental conditions. So, the main difference between acclimation and adaptation is that acclimation occurs within the lifetime of an organism, whereas adaptation occurs over several generations. Furthermore, acclimation allows organisms to adjust their morphological, behavioral, physical, or biochemical traits in response to changes in the environment. Meanwhile, what adaptation does is that adaptation allows organisms to fit their environment by enhancing their evolutionary fitness. In addition to these, acclimation does not produce a novel phenotypic trait, hence it does not proceed through natural selection. Adaptation, on the other hand, produces a novel phenotypic trait that has a functional role in the environment, and therefore it proceeds through natural selection. So in brief, acclimation and adaptation are two processes that allow organisms to change their characteristics according to the environment. Both processes enhance the life of the organism. So, what is acclimation? Acclimation or acclimatization is the process by which an individual organism adjusts its characteristics according to its environment. Usually, these changes occur suddenly, either due to the migration of the organism to a new habitat, such as changes in the altitude due to changes in the climate, for example, temperature, humidity, etc., changes in pH, photoperiod, etc. Moreover, the main function of acclimation is to allow the maintenance of the performance of the organism across a wide range of environmental conditions. Furthermore, organisms may change their morphological, behavioral, physical, and biochemical traits in response to changes of the environment. For example, organisms acclimatize in response to the changes in the environment by changing the biochemistry of the cell membrane. Basically, this is by increasing the number of membrane proteins as well as by expressing heat shock proteins. It makes the cell membrane more fluid in cold temperatures and less fluid in warm temperatures. What is adaptation? Adaptation is a dynamic evolutionary process which helps to fit the organism to their environment by enhancing their evolutionary fitness. And it occurs in a population of species but not in an individual organism. Also, it produces a new phenotypic trait with a functional role. Furthermore, the evolution of novel phenotypic trait occurs through natural selection. So, moreover, adaptation increases the biological fitness of a population or species by altering the genetic content of the genome. Therefore, the rate of evolution is measured by the change in gene frequencies. In addition, two or more species can co-evolve by developing adaptations to the same environment. For example, flowering plants and pollination insects. Furthermore, mimicry is an excellent example of adaptation of a species to a particular environment. It is responsible for evolving for an evolving species resembling an object in the environment or to an organism. There are also some similarities between acclimation and adaptation. These two processes are responsible for changing characteristics of organisms according to the environmental conditions, very evident. Also, they facilitate the living of organisms. There are differences which is more prominent. Acclimation will refer to the gradual reversible change of a body of an organism to better suit the changes of the environment, while adaptation refers to the process by which a species becomes more fitted to its environment as a result of natural selection, acting upon heritable variation over several generations. Time taken. Acclimation occurs within the lifetime of the organism, while adaptation occurs over several generations. Type of response. Acclimation is a short-term response to changes in the environment, while adaptation is a long-term response. Type of change. Acclimation allows organisms to adjust their morphological, behavioral, physical, and biochemical traits. As I already said, adaptation allows organisms to fit the environment through enhancing their evolutionary fitness. Occurrence. Acclimation occurs when an organism gets the use of a new environment by migration or due to the changes in the climate in their new habitat. Adaptation broadly applies to a population or species as an essential aspect of the evolutionary process. Types of organisms. Acclimation occurs in individual organisms when adaptation occurs either in a population or species. Genetic changes. Acclimation does not really bring in genetic changes, adaptation brings genetic changes. Production of a phenotypic trait. Acclimation does not produce a novel phenotypic trait, while adaptation produces a novel phenotypic trait through mutations. Then natural selection. In addition, acclimation does not really proceed through natural selection, while adaptation proceeds through natural selection. Acclimation can occur within hours or weeks, while adaptation can be measured through changes of gene frequencies. Acclimation is a reversible process, while adaptation is an irreversible process. Acclimation does not really contribute to evolution, while adaptation contributes to evolution. So, furthermore, acclimation is the adjustment into altitude, temperature, humidity, photoperiod, pH, while mimicry is an example of adaptation. So, this the main conclusion is that the acclimation does not bring transient changes to the organism. These changes do not proceed through natural selection in order to produce a new phenotype type. Adaptation is another type of change which occurs through generations, which occurs to populations or population and not exactly to a particular organism. There is a difference between acclimation and adaptation. Is the type of change in the environment, and the type of change is produced by organisms in response to the changes. So this is the basically difference between adaptation and acclimation. So this slide also summarizes the difference between adaptation and acclimatization or acclimation. Anyway, you're right. This is the answer is correct. The fourth option will be correct. Moving on to the next question. This This is also another slide that talks about the difference between both the processes occurs within lifetime of an organism, several generations, short term, long term, uh, individual organisms, population or subspecies, genetic changes does not bring genetic changes, brings genetic changes, phenotypic trait can occur really fast, it's very slow, reversible, irreversible, etc. Moving on to the next question. Trophozoites of Cryptosporidium are embedded in anybody? Anybody knows the answer? Trophozoites of Cryptosporidium. Think if you know the answer. These are really theoretical questions, not really conceptual. So you have to know it to answer it. You cannot think about it at the instant and answer it.
Option 2.
Human gastrointestinal epithelium. Anybody else? So you're correct. It's human GI, gastrointestinal epithelium. So what are tropozoites and what is cryptosporidium basically? So many species and genotypes of this apicomplexan protozoan called cryptosporidium species can infect humans, and they have a wide range of host animals. So zoonotic species and genotypes of cryptosporidium are those that are transmitted from animal hosts to humans, and non-zoonotic species and genotypes that the host adapted without evidence of transmission from animals to humans. So Cryptosporidium parvum, which was formerly known as Cryptosporidium parvum genotype 2, and Cryptosporidium hominis or hominis, formerly known as Cryptosporidium parvum genotype 1, are the leading causes of human cryptosporidiosis. Cryptosporidium meligridis, Cryptosporidium felis, Cryptosporidium sporidium canis, Cryptosporidium ubiquitum, Cryptosporidium corniculus, Cryptosporidium viatorum. All these can also infect humans. There are a lot of other variants also. So what happens is that sporulated oocytes containing 4 sporozoites are excreted by the infected host through species. Now through this feces and possibly other routes such as respiratory secretions. Now transmission of the Cryptosporidium species occurs mainly through ingestion of the fecally contaminated water, like this drinking or recreational water or food like raw milk, or the following like or direct contact with the infected animals or people. So following ingestion and possibly inhalation by a suitable host is how the recreational water and these things are happening in this is in the feast. Now feces. So following ingestion and possible inhalation by a suitable host, excistation occurs basically. Now what is excistation? The sporozoites are released, and these parasitize the epithelial cells of the gastrointestinal tract and possibly the respiratory tract. Now, in these cells, usually within the brush border, the parasites undergo asexual multiplication, which is called schizogony or merogony, and then sexual multiplication, which is called schizogony or merogony, gametogony. Sorry, sexual multiplication is called gametogony, producing microgametes like male, and macrogametes which are female. Upon fertilization of the macrogametes by the gametes that rupture from the microgamete, oocysts develop and sporulate in the infected host. And zygotes give rise to 2 different types of oocysts, thick-walled and thin-walled. Thick-walled oocysts are excreted from the host into the environment, whereas the thin-walled oocysts are involved in the internal autoinfective cycle and are not recovered from stools. And oocysts are infectious upon excretion basically, this enabling direct and immediate fecal oral transmission. And extracellular stages have been reported, but the relevance in the overall life cycle is still very unclear. So, Cryptosporidium can infect a wide range of vertebrate hosts, including birds, reptiles, and mammals, and many species and genotypes are also there, host adapted. But human cases caused by species and genotypes that are pathogens in other mammals or animals have also been reported, such as Cryptosporidium meliagridis, and predominantly which infects humans and is generally considered anthroponotic, though sporadic reports in animal hosts also exist. And zoonotic subtype families of Cryptosporidium parvum implicated in human infections are commonly associated with the cattle, particularly calves. So, what is the geographic range? Zoonotic and non-zoonotic Cryptosporidia species and genotypes are ubiquitous worldwide. So, outbreak of cryptosporidiasis have been and continue to be reported in several countries. And cryptosporidiasis outbreaks in the US have been linked to swimming pools, water playgrounds, and other swimming venues. Unpasteurized cider, juice, milk, contact with animals, childcare settings, camps, ill food handlers, and what is the clinical presentation? Like infection with cryptosporidium species and genotypes, that's in a wide range of signs and symptoms. Like the incubation period is an average of 7 days, ranges from 2 to 10 days, and immunocompetent patients may present with diarrheal illness that is self-limiting, typically resolving within 2 to 3 weeks. However, immunocompromised patients already suffering from any other diseases may have more severe complications, such as life-threatening malabsorption and wasting. Like diarrheal illness may be accompanied by fever or fatigue. So while the small intestine is primarily affected, extraintestinal cryptosporidiosis, that is example in the pulmonary or in the biliary tract, rarely in the pancreas, has also been reported. So the tryptozoites of the cryptosporidium are very embedded in the human gastrointestinal epithelium. So the answer is correct. Moving on to the next question. Schistosomiasis is diagnosed through the detection of parasite eggs in urine, blood, biopsy sample, cerebrospinal fluid. Anybody?
Blood.
Okay. Anybody else like to try? It's actually urine. So before that, we'll learn some of the key facts about schistosomiasis. So, what are the key facts? Like lack of hygiene and certain play habits of school-age children, such as swimming or fishing in infested water, actually make them especially vulnerable to this infection. So, in 2021, when the COVID pandemic was going on and work was being performed to mitigate its impacts, it decreased the provision of neglected tropical diseases, interventions, and the treatment of coverage for schistosomiasis. Sorry. So it's an acute and chronic disease caused by the parasitic worms, and people are infected during routine agricultural, domestic, occupational, and recreational activities, which exposes them to infested water. And estimates show that at least 251.4 million people required preventive treatment from schistosomiasis in 2021, out of which more than 75.3 million people were reported to have been treated. So, pseudosomiasis control focuses on reducing disease through periodic large-scale population treatment with, a drug called, I think, Prasvintical. I'm not able to pronounce it correctly. And there's a more comprehensive approach also, including potable water, adequate sanitation, and snail control would also reduce, transmission. So, what is schistosomiasis actually? It is basically acute and chronic disease caused by the blood flukes, which are the trematode worms of the genus Schistosoma. And estimates show that at least, like as I said, 251.4 million people require treatment. and this preventive treatment, which should be repeated over a number of years, will reduce and prevent morbidity. So this transmission has been reported for over 78 countries. However, preventive chemotherapy for schistosomiasis, where people and communities are targeted for large-scale treatment, is only required in 51 endemic countries with moderate to high transmission. So how does the infection and how does the transmission happen? So people become infected when larval forms of the parasite released by freshwater snails penetrate the skin during contact with infested water. Transmission occurs when people suffering from schistosomiasis contaminate freshwater sources with feces or urine containing parasite eggs which hatch in water. Very prevalent in the rural areas in India till today. And in the body, the larvae develop into adult schistosomes. And adult worms live in the blood vessels where the females release eggs. Some of the eggs are passed out of the body in the feces or urine to continue the parasite's life cycles. So others become trapped in the body tissues, causing immune reactions and progressive damage to the organs. If you think about epidemiology, it's very prevalent in tropical and subtropical areas, especially in poor communities without any access to safe drinking water and adequate sanitation. It's estimated that at least 90% of those requiring treatment for schistosomiasis live in Africa. And there are actually two major forms of schistosomiasis: intestinal and urogenital, caused by five main species of blood fluke. The intestinal schistosomiasis is caused by Schistosoma mansoni, Schistosoma japonicum, Schistosoma mekongi, Schistosoma guinearis, and Schistosoma intercalatum. Mostly prevalent in China, Philippines, Africa, Brazil, Venezuela, and all these areas, rainforest areas of Africa. The urogenital schistosomiasis is very prevalent. It's caused by Schistosoma hematobium, very prevalent in Africa, Middle East, Corsica, France. It mostly affects poor and rural communities, particularly agriculture and fishing populations. And women doing domestic chores in infested water, such as washing clothes, are also very typically at risk and can develop female genital schistosomiasis. So, inadequate hygiene and contact with infected water make children especially very vulnerable to this infection. So migration to urban areas and population movements are introducing the disease to new areas as well. So increasing population size and the corresponding needs for power and water often result in these development schemes, and environmental modifications facilitate transmission. So with the rise in ecotourism and travel to remote areas, increasing numbers of tourists are contracting schistosomiasis. So at times, tourists present severe acute infection, and unusual problems include paralysis also. So, and urogenital schistosomiasis is also considered to be a huge risk factor for HIV infection, especially in women. So, the symptoms are caused mainly by the body's reaction to the worm's parasite's eggs. So, intestinal schistosomiasis can result in abdominal pain, diarrhea, blood in the stool. Liver enlargement is common in advanced cases and is frequently associated with an accumulation of fluid in the peritoneal cavity and the hypertension of the abdominal blood vessels. And in such cases, there are also may be enlargement of the what's that called, spleen, enlargement of the spleen as well. So the classic sign of urogenital schistosomiasis is hematuria, like blood in urine. So it's a very important symptom when you see there's blood coming out of urine. And kidney damage and fibrosis of the bladder and the ureter are sometimes diagnosed in very advanced cases. Bladder cancer is another possible complication in later stages. In women, urogenital schistosomiasis may present with genital lesions, vaginal bleeding, pain during any kind of intercourse, nodules in the vulva. And in men, urogenital schistosomiasis can induce pathology of the seminal vesicles, prostate, and other organs. So, the disease may also have other long term irreversible consequences, including infertility. And the economic and health effects of schistosomiasis are considerable, and the disease disables more than it kills. So in children, schistosomiasis can cause anemia also, growth stunting, and a reduced ability to learn, although the effects are usually reversible with treatment. But chronic schistosomiasis may affect people's ability to work, and in some cases can even result in death. So the number of deaths due to schistosomiasis is difficult to estimate because of hidden pathologies such as liver, kidney failure, bladder cancer, ectopic pregnancy due to female genital schistosomiasis, and all these. So I think deaths due to schistosomiasis are currently estimated at 11 to 12,000 globally per year. But these figures are probably underestimated and they need to be reassessed. So how do you diagnose this kind of this disease, but which was the main question. So schistosomiasis is diagnosed through the detection of parasite eggs in the stool or the urine specimens. That's why urine will be the right answer. Basically, in the stool or the urine specimens, antibodies and antigens are detected in the blood or urine samples are also indications of infection. So in the blood also you can get some kind of antibodies, but parasite eggs, if you want to check the detection of parasite eggs, then that will be either urine or stool. In blood, if you want to check, you can actually find some antibodies when the body is trying to fight the disease. For urogenital schistosomiasis, a filtration technique using nylon, paper, or polycarbonate filters is a standard diagnostic technique. So children with schistosomiasis hematobium almost always have microscopic blood in the urine, which can be detected by chemical reagent strips. So the eggs of the intestinal schistosomiasis can be detected in fecal specimens through a technique using methylene blue stained cellophane soaked in glycerine or glass slides, known as the cat to cats technique. So in cystosoma mansoni technique transmission areas, the circulating cathodic antigen test can also be used. And for people living in low endemic or low transmission areas, serological and immunological tests may be useful in showing exposure to infection and the need for thorough examination, treatment, and follow up. So how can this disease be prevented, or what are the controls? So it's the control is basically based on large scale treatment of at risk population groups, access to safe water, improved sanitation, hygiene education, behavior change, cell control, and environmental management. So then, like the new neglected tropical disease roadmap of 2021-2030, adopted by the World Health Assembly, set as global goals the elimination of schistosomiasis as a public health problem in all endemic countries and the interruption of its transmission in selected countries. So the WHO strategy for schistosomiasis control focuses on reducing disease through periodic targeted treatment with praziquantel, I think that's the name, praziquantel, through large-scale treatment of or preventive chemotherapy of affected populations. So it involves regular treatment of all the at-risk groups, and in few countries where there is low transmission, the interruption of the transmission of the disease should be also be aimed for. So the groups that should be targeted at first for treatment are preschool children, then school age children, adults considered to be at risk in endemic areas, and people with occupations involving contact with infested water, such as fishermen, farmers, irrigation workers, women whose domestic tasks bring them in contact with infested water, such as washing clothes and utensils. So entire communities in highly endemic areas. So WHO recommends treatment of this infected preschool and age children based on the diagnostic and the clinical judgment, and the inclusion in the large scale treatment using praziquantel formulation. So the aim is to reduce disease morbidity and transmission through the elimination of the disease as a public health problem. And praziquantel, I think, is the recommended treatment against all forms of schistosomiasis. Sorry, it is effective, safe, and very not very low cost. Even though reinfection may also occur after treatment, the risk of developing severe disease is diminished and even reversed when treatment is initiated and repeated in childhood. And this schistosomiasis control has been widely implemented over the past 40 years in several countries, including Brazil, Cambodia, China, Egypt, Mauritius, the Islamic Republic of Iran, Oman, Jordan, Saudi Arabia, Morocco, Tunisia, and all these places. And in many countries, it has been possible to scale up schistosomiasis treatment to the national level and have an impact on the disease in a few years. So over the past 10 years, I think there has been a scale up of treatment campaigns in a number of sub-Saharan countries where most of those are at risk life. So these treatment campaigns resulted in the decrease of prevalence of schistosomiasis in school-age children by almost 60%. So yeah, the answer will be stool or urine. And the diagnosis of schistosomiasis, stool and urine are the most commonly used clinical samples for DNA isolation. Other biofluids such as serum, saliva, and cerebrospinal fluid are also used, particularly for cell-free DNA detection assays. Like conventional DNA extraction methods include precipitation techniques with phenol, chloroform, and ethanol isopropanol. However, the feasibility of applying these methods has in the past been affected by the potential for direct exposure of hazardous chemicals to operators and the significant time involved. However, the improved However, the improved quality and yield of isolated DNA has also helped. The protocols involve techniques basically such as spin column based purification and magnetic based DNA separation. Furthermore, these kits have been developed for use on different clinical samples types to overcome the unique challenges related to each. And moreover, recent advances in DNA purification technology have resulted in high throughput automated platforms, minimizing the time spent, the labor involved, and manual handling, making the process convenient, efficient, and accurate. However, as is common to most high-tech applications, these automated techniques involve costly equipment and are currently not really suitable for field application in disease endemic resource poor settings. And most DNA amplification methods generally need to be performed in well-equipped centralized facilities that are generally located distant from the clinical specimen collection site. So therefore, under these circumstances, it is important that feasible and rapid sample preservation methods are available prior to DNA extraction and amplification. So, potential solutions to these issues include the preservation of critical specimens such as finger prick, blood, urine, or stool on filter papers as dried spots for the convenient transportation to a central lab for DNA extraction and application or direct PCR. So, this is all about schistosomiasis. Moving on to the next question. And antibiotic resistance, option A, not option statement A, antibiotic resistance can be transferred from one pathogenic cell to another through extrachromosomal genetic material like plasmid. Also, antibiotic resistance cannot be transferred horizontally and vertically. Which option is true? Anybody? Anybody? Try.
Both statements are correct.
Anybody else? So both statements are not correct. Antibiotic resistance can be transferred horizontally. Statement A is correct. Statement B is not correct since it mentions cannot. So this statement is not correct because antibiotic resistance can be transferred horizontally via horizontal gene transfer techniques. So what happens is though the harnessing of antibiotics is one of the most significant human innovations, the efficacy is continuously eroded by the craftiness of the microbial targets. So once a single bacterium mutates to become resistant to antibiotics, it can transfer that resistance to other bacteria around it through a process called horizontal gene transfer. Okay, is there a better image? No, okay. Horizontal gene transfer. So one of the main vehicles for gene transfer among bacteria are small circular pieces of DNA or plasmids. Plasmids can be transferred through direct physical contact between bacteria in a process known as conjugation, which helps bacteria share their antibiotic resistant genes with their neighbors. Now, although conjugation is well understood on a molecular level, how it plays out in the environment that bacteria really inhabit rather than in the lab is much less clear. So, one especially versatile gastrointestinal pathogen called Salmonella enterica serover typhimurium is particularly interesting for studies on resistance gene sharing because it forms so-called persister reservoirs in its host. And in these cases, antibiotic resistant cells lurk in the gut tissue or other organs after an infection and migrate back into the gut lumen to cause reinfections after the antibiotic pressure is gone. So how does this conjugation occur? So there are helper plasmids also which facilitate conjugation. So as Salmonella typhimurium is frequently encounters gut bacteria, plasmid sharing and the spread of resistance genes is a real concern. So there was a study that was recently published in ASM's Journal of Bacteriology, which discovered that a particular strain of Salmonella typhimurium known as SL1344, it shares its plasmid with other bacteria using the assistance of another plasmid. The study was led by researchers at the ETH Zurich in Switzerland, and it focused on a plasmid that encodes resistance genes for streptomycin and sulfonamides, called P3 for short. However, this P3 it lacks the genes for the conjugation machinery itself, meaning it needs a helper plasmid to move between the cells. In Salmonella typhimurium, this helper plasmid is known as P2. So on a sequence level, P3 very closely resembles another plasmid which has a broad host range, meaning it can replicate in a wide variety of bacterial species. So the researchers hypothesized that P3 may be transferred from Salmonella typhimurium to diverse bacterial species in the mammalian gut environment, potentially spreading antibiotic resistance genes as it goes. The hypothesis was also tested using mice, because the mice were first infected with one of the recipient bacterial species, including representatives of the human gut flora, then with Salmonella typhimurium 24 hours later. The researchers then monitored the growth of both the recipient and Salmonella typhimurium as well as the frequency of P3 transfer by analyzing the phases of the mice over 3 days. They identified P3 transfer mediated by P2 occurring between Salmonella typhimurium and 4 recipients belonging to the class Gamma proteobacteria, I guess, representing the gut commensals as well as plant associated bacteria. So although this P3 plasmid appears to be very shareable among diverse bacteria within and beyond the mammalian gut, however, the researchers did not expect the plasmid to be transferred if there was no antibiotic pressure, as it would not directly benefit the bacteria to harbor resistance genes. They were surprised by their findings. They said that the most striking finding was that the plasmid was taken up by other bacteria even without the selective pressure of antibiotics. So there's this plasmid paradox also, that it still remains unclear whether P3 persists in its recipients long term and why it was transferred in the first place, even when it did not directly benefit the bacteria. So this is known as the plasmid paradox, to which several solutions have been proposed. For example, the plasmid may carry unknown fitness benefits besides antibiotic resistance. So indeed, a recent study using another broad host and resistant bacteria, host resistant plasmid, showed that it can have a wide range of effects across different recipient strains, with some gaining a fitness benefit by maintaining the plasmid. So alternatively, the plasmid may also be acting as a purely selfish DNA, only concerned with their own persistence and replication. So conjugative plasmid transfer has also been found to be perpetuated by non-antibiotic pharmaceuticals such as certain painkillers and beta blockers. So in a 2022 study, researchers discovered that common drugs such as ibuprofen and propanolol can boost the transfer of broad-range multi-resistance plasmid (RP4) from Pseudomonas putida to phylogenetically diverse bacteria in activated sludge. And the researchers also show that overproduction of reactive oxygen species by bacteria in the presence of pharmaceuticals likely contributed to this enhanced conjugative activity. So, how do you manage this AMR crisis? AMR is antimicrobial resistance crisis. So, best practice guides urge the reduced use and abuse of antibiotics in both clinical and agricultural settings in order to reduce the selective pressure for transfer of resistance genes. Therefore, the spread of plasmids such as P3 and RP4 in the absence of this pressure is concerning, as it suggests that reducing antibiotic use in population may not be enough to curb this rising resistance. This means that even antibiotic resistance can spread even when antibiotics are not involved. Altogether, studies that probe the mechanisms and dynamics of plasmid transfer among bacteria are vitally very important. So, by understanding where, how, and how often plasmids are shared, they can continue to seek and develop solutions for emerging multi-drug resistant pathogens, as well as quantify risks and manage the world's ever-growing populations of antibiotic resistant pathogens. So what's more is that they can also understand what makes a plasmid shareable and even how to stop conjugation from happening to curb the spread of antibiotic resistance. So basically, as you know about plasmids, that the bacterial chromosome is made up of DNA, and it stores all the information that a bacterial cell needs to carry in normal function. In addition to the chromosome, bacteria can have small circles of DNA called plasmids that contain genes. The antibiotic resistance genes were not common in plasmids before the introduction of antibiotics as medicines. Today, however, genes that provide protection against different antibiotics as well as resistance to, for example, heavy metals like copper, silver are very commonly found under the same plasmids. One of the antibiotics or metals is then enough to maintain or select for all the antibiotic resistant mechanisms since they are linked together on the plasmid. And this phenomenon is known as co-selection of resistance genes. And plasmids can also easily transfer between different bacteria. Plasmids also often have mechanisms for the transfer of the whole plasmid to other bacteria. This means that a bacteria the bacteria can become resistant to multiple antibiotics at once by picking up a single plasmid. They then become multi-drug resistant. Furthermore, genes that influence bacterial virulence can also frequently be found on plasmids, and pathogenic bacteria carrying these multi-drug resistance plasmids have increased in prevalence and now cause major problems around the world. So there are very few treatment options remaining for these infections. Co-selection is one of the reasons for why it is so difficult to reverse this resistance once it has been established in a bacterial population. It's simply not enough to stop using one particular antibiotic if the resistance mechanism is linked to resistance for other antibiotics as well. So even that's why doctors ask you, like if you are using an antibiotic, they ask you to finish the antibiotic dose. People generally people take antibiotics without consulting doctors, which is very very wrong. And then people start taking antibiotics and then they stop before the dose is finished. So if the dose is not finished, it's very easy for the bacteria inside you to become resistant to the antibiotic, and then they'll keep on transferring this resistance to other bacteria. This is how multi-drug resistant TB, Mycobacterium tuberculosis has also come into existence. So there are so many bacteria that have become drug resistant because of this practice led by humans, where they do not consult professionals. They try to use their own brain, and they intake a lot of antibiotics without consulting any professional doctor. They stop the dose in between, very wrong. So definitely, before starting any antibiotic dose, definitely consult a doctor. And even if the doctor has recommended antibiotics, you have to finish the entire dose. Don't stop in between. That leads to massacre, because the bacteria inside you, it gets resistant to the antibiotic, and then it can very easily transfer its antibiotic resistant gene to other bacteria. So the first statement is true. Second statement is not true. So only statement A is correct. That is how this question can be answered. Moving on to the next question. During elongation step in PCR, forward primer is of utmost importance as the polymerization will start from this point. However, addition of reverse primer is optional. True or false? Anybody?
True.
It's true. We can do PCR even without adding reverse primer.
No, so it is false.
Which part of the sentence is false? Which part is true? Which part of the sentence is false? Others can also try. We are all learning. Like, why do you think it's true and why do you think it's false? Then I'll let you know if reverse primer is optional or not. So the answer is actually false. Reverse primer is not optional. You need both forward and reverse primers for doing PCR,
Okay.
and like only forward primer will never help, because reverse primer is also equally important, and that we'll come to know, like that we can discuss when we talk about polymerase chain reaction. So how does this thing exactly happen? So polymerase chain reaction is commonly known as PCR, and it's used to produce multiple copies of a gene or DNA of interest. And the steps of PCR are denaturation. First is denaturation, primer annealing, and extension of the primers. This is one of the revolutionary discoveries. This technique is one of the revolutionary discoveries in the history of science, like discovering that DNA is genetic material and being able to amplify a particular uh, genetic particular gene or a particular DNA fragment. This technique is revolutionary. I don't think biology would have progressed this far without this technique. So that's why even when you're studying, we should always appreciate how this technique came into existence. In today's time, we can read about it and we can think it's very simple, but when this technique came into existence, the way the scientists who actually came up with this technique, it's immaculate. It was PCR was invented by Carey Mullis in 1983. Imagine thinking like this in 1983, with such limited resources, with such limited knowledge about everything, and still discovering the technique. It's very fascinating. So with this technique, you can produce multiple copies of a gene or DNA of interest. So what happens? It's a reaction. You can amplify a gene. So it is done in vitro, like in test tubes, using a primer. And this technique is used in labs to make billions of copies of the desired gene for research, diagnostic, and therapeutic purposes. As I said, this was invented by Carey Mullis in 1983, and it requires a DNA primer. This red one is a primer designed for the DNA template, and a DNA polymerase. The DNA polymerase is an enzyme. It's not shown here. So there's a DNA polymerase. It's an enzyme, which is preferably a very thermostable DNA polymerase, an enzyme which can stay stable even at high temperatures. And in the PCR reaction, the reaction cycle is repeated many times to get billions of copies. So it's an integral part of biotechnology, medical biology, diagnostics, forensic analysis, molecular biology research, etc. And the amplified DNA can be sequenced, cloned, and visualized by gel electrophoresis. So this is polymerase chain reaction. Each cycle of the polymerase chain reaction has 3 steps. They are denaturation. So in the first step, denaturation, this step is required. So we know DNA is double-stranded. Denaturation is required to separate the double-stranded DNA sample, and it is done at typically above 90 degrees Celsius, 94 to 98 degrees Celsius for 20 to 30 seconds. So it breaks the hydrogen bonds between the base pairs. We know that adenine, thymine have 2 hydrogen bonds in between them. Guanine and thymine have 3, GC have 3 hydrogen bonds. So at this temperature, the hydrogen bonds are broken between the base pairs, and denaturation leads to the formation of single strands of DNA. So this is a single strand, this is a single strand. Then comes the annealing step. So this second step is called the annealing. And why is it called annealing? Because primer is a sequence that is complementary to one end of your gene of interest. So the annealing occurs, and here the reaction temperature is lowered to allow the complementary base pairing. The reaction temperature is lowered, and the complementary base pairing is allowed between the primer and the complementary part of the single strand of the DNA template. And a proper temperature needs to be maintained in order to allow highly specific and proper primer hybridization. Then DNA polymerase binds to the template primer hybrid and starts the DNA synthesis. Then extension. So a thermostable DNA polymerase is used for this purpose. Taq polymerase is commonly used for this purpose. It is done at a temperature of 75 to 80 degrees Celsius, 72 degrees Celsius. The DNA polymerase adds nucleotide in the 5' to 3' direction, very important, and synthesizes the complementary strand of the DNA. So this is how the complementary strand is formed. And this entire cycle is repeated 25 to 30 times, and with this the DNA sample can be amplified a billion times. After this, gel electrophoresis is used to visualize the result of the PCR. So PCR reaction requires a primer, thermostable DNA polymerase, template DNA, and nucleotides. So what is the role of the primer for polymerase chain reaction? Because the question was related to primer. So 2 sets of primers are used, which are short stretches of oligonucleotides. They are chemically synthesized and complementary to the part of DNA template to be amplified. So DNA polymerase requires a primer for initiating replication. DNA polymerase cannot initiate the process, it can only add nucleotides. So therefore a primer is required uh, uh, like required for this purpose to initiate the polymerization process. Primers are usually 20 nucleotides long. The two primers used, the two primers used are called forward primer and reverse primer. They are usually 20 nucleotides long, and they cover the target region of the DNA to be amplified and bind to the opposite edges of the template strand. You see, opposite edges template strand. So after binding of the primers, the region between them gets copied by the DNA polymerase. Now, what is a Taq polymerase? Taq polymerase, basically the replication of the polymerization of DNA requires a DNA polymerase enzyme. So, in PCR, Taq polymerase is used because it is a very thermostable enzyme, and it is isolated from a heat tolerant bacteria called Thermus aquaticus found in Yellow Springs. A thermostable polymerase is required so that it can remain active even at very high temperatures. So higher temperatures are used in the PCR process because for denaturation you require a very high temperature. So Taq polymerase is most effective at around 70 degrees Celsius, which works fine for this process. So this PCR technique is has many advantages. It's a highly sensitive technique. It gives results rapidly. It's simple, easy to understand. It's highly specific, important tool in microbiology, biotechnology. Millions and billions of copies of the desired gene can be produced, which can be utilized for various purposes such as sequencing, analysis, cloning. It can be used for diagnosis. A viral genome can be amplified and detected for the presence in the host cell. A genetic sequence associated with a bacterial toxin can be diagnosed efficiently using PCR. PCR amplification enables the detection of a viral genome early after infection. It can help in diagnosing the infection even before the onset of the disease. Viral load can be detected. RT-PCR is commonly used to detect the SARS-CoV-2 viral genome. It is a variant of PCR and uses the enzyme reverse transcriptase to produce DNA complementary to RNA. Then oncogene mutations can be detected very early with PCR. It helps in diagnosing various cancers such as leukemia. PCR is used in forensics. A small sample of DNA can be collected from the crime scene, can be amplified and analyzed and compared with the suspects. PCR can be used for genetic testing, microbiology, molecular biology research, recombinant DNA technology, useful in detecting and analyzing ancient DNA samples. It's helpful in analyzing phylogenetic relationships. It's also used to study gene expression. It can be used to create mutant genes of choice to study site-related mutagenesis. Basically, it's a very important tool, quantitative PCR or qPCR. So the two primers that are used, forward primer and reverse primer, are used in each PCR reaction, which are designed to flank the target region for amplification. Now, two complementary DNA strands, single strands of DNA are released during denaturation. The forward primer binds to the template DNA while the reverse primer binds to the other complementary strand, both of which are amplified in the PCR reaction. If only one, what would happen if only one primer is added? If only one primer is used, the process is called asymmetric PCR. So only one strand of the DNA, double-stranded DNA, will be amplified, and only one new copy is synthesized per cycle, which is unable to achieve exponential amplification. So that is why reverse usage of reverse primers is not optional; it is mandatory for PCR to occur properly. I hope that the doubt has been clear. So that's why this statement is entirely false. The first part is still fine. Forward primer is of utmost importance. Addition of reverse primer is optional. This statement is wrong. So that's why this entire statement is false. Moving on to the next question. In TACMAN PCR, forward and reverse primers are not required as the probe independently gets the job done. We'll discuss what TACMAN PCR is, but before that, anyone wants to try?
No idea, man.
Anybody else? Okay, we'll discuss what a TACMAN PCR is, and then we'll talk about primers and probes. So the answer is false. So what is a TACMAN PCR basically? Anybody has any idea about TACMAN PCR? No idea about TACMAN PCR? Okay, so TACMAN PCR is basically a type of real-time PCR. It uses a nucleic acid probe complementary to an internal segment of the target DNA. So this is the TACMAN probe. It is complementary to the internal segment of the target DNA. So the probe is labeled with two fluorescent moieties fluorescent moieties. The emission spectrum of one overlaps with the excitation spectrum of the other, resulting in quenching of the first fluorophore by the second. So this is called R skew. So one of the emission spectrum of one overlaps with the excitation spectrum of the other. So it basically quenches the first fluorophore by the second. So if these two fluorophores are very close in vicinity, then this will quench this. So the probe is present during the PCR, and if the product is made, the probe is degraded by the 5' nuclease activity of Taq polymerase that is specific for DNA hybridized to the temple. The degradation of the probe allows the two fluorophores to separate. So the forward primer is here, reverse primer is here. The DNA polymerase will attach here, and it will help in elongating the primer to form the complementary strand. When it reaches this point, the polymerase, the DNA polymerase has another activity. It has a 5' nuclease activity. Nuclease means it degrades nuclear degrades basically genetic element. So it has a 5' nuclease activity. So what it does is that it degrades this probe. When it reaches the probe, has hybridized to this part. So when the forward primer has elongated till here, the DNA polymerase will be here. It will be close to the TACMAN probe. It will degrade the TACMAN probe because of its 5' nuclease activity, and the degradation of the probe will allow these two fluorophores to separate. So when this probe is very small, so at this point of time these two fluorophores are very close, and so this element is this Q is quenching the fluorescence of R. But when DNA polymerase comes here, it degrades this probe. These two get separated. So now R and Q are no longer very close. So now there is no quenching happening. So these two fluorophores separate. This reduces quenching and increasing intensity of the emitted light. So R starts emitting. And because this assay involves fluorescence measurements that can be performed without opening the PCR tube. So inside the PCR tube, this fluorescence measurement can be happening. The risk of contamination is also reduced. You're not adding anything from outside. Everything is happening inside the PCR tube. The risk of contamination is also reduced. So this is also known as the 5' nuclease assay, because TACMAN probes are labeled with two fluorescent dyes and emit at different wavelengths. The probe sequence is intended to hybridize specifically in the DNA target region of the interest between the two PCR primers. Typically, the probe is designed to have a slightly higher annealing temperature compared to the PCR primers, so that the probe will be hybridized when extension of the primers begin. A minor groove binder is sometimes used near the 3' end of the TACMAN probe to enable the use of shorter sequences that have higher annealing temperatures that would be expected for sequences of equivalent length. The reporter dye is attached to the 5' end of the probe sequence, while the quencher dye is attached on the 3' end. A popular combination of dyes can be the FAM or the Vic dye, or the reporter dye and the Tamra for the quencher dye. So when the probe is intact and the reporter dye is in close proximity to the quencher dye, little to no fluorescence will result, because of the suppression of the reporter uh, due to the an energy transfer between the two dyes. During polymerization, strand synthesis will begin to displace any TACMAN probes that have hybridized to the target sequence. The Taq DNA polymerase used has a 5' exonuclease activity, and therefore will begin to chew away any sequences in its path, that is, those probes that have annealed to the target sequence. So when the reported dye is released from the probe and is no longer in close proximity to the quencher dye, it can begin to fluoresce. An increase in the fluorescent signal results in the target results if the target sequence is complementary to the TACMAN probe. It is important to note that the mismatches between the DNA template and the TACMAN probe can cause failure to detect the DNA template appropriately. So some assays known as the quantifier kit include an internal PCR control, or the IPC also, that enables verification that the polymerase, the assay and the detection instrumentation are working properly. So in this case, the IPC is labeled with the Vic reported dye and hybridizes to a synthetic template added to its reaction. The TACMAN probe for detecting the target region of interest is labeled with a FAM. Dye is therefore specifically spectrally resolvable from the green Vic dye. And there can be other instruments such as which can be well placed in each well to adjust well to well differences across a plate through background subtraction also. So TACMAN is a fluorogenic probe based PCR assay in which situated between the two PCR primers is an internal oligonucleotide probe with a fluorescent label attached at the 5' end and a quenching molecule attached at the 3' end. And the internal oligonucleotide hybridizes to the template, is digested by the 5' to 3' end nucleus activity of the thermus aquaticus or tagged DNA polymerase. When the PCR primer is extended, it's digested only if so this thing will get digested only if DNA replication is properly occurring, separating the fluorescent and the quencher molecules. So PCR products can be Thank you. No problem. So nobody has any question. We can maybe conclude the session and we can talk. We can meet next session next Friday, same link, same time. Okay. Thank you so much. I'll stop the meeting.
Thank you, ma'am.
Thank you.